Good morning, good afternoon, everybody. Thank you for attending today's session for Clinical Trial Digital Innovation Patient-Centric Sampling. Today's presenters are Dr. Olga Kubetska from Pfizer and Dr. Deanne Jackson-Rudd from Merck. People who don't know me, my name is Shifali Patel. I work at Janssen Pharmaceuticals, and I will be the moderator for this session. Before I introduce our speakers for today, I would like to show this slide, which shows our current patient-centric working group. As you can see, we have people from Translational and ADME leadership group as well as clinical leadership group from different organizations who form this team. This team was formed earlier this year in 2020, and our chairs, members, as well as sponsors are shown on this slide. Without further ado, I would like to introduce our speakers for today, Dr. Olga Kubetska, who is Global Head for Clinical Assay Group at Pfizer. Olga graduated from Chemistry Department of Moscow State University. Olga spent over 20 years of her career with pharmaceutical industry, leading and managing regulated bioanalytical teams at CRO, Pfizer, and AbbVie. In last 5 years, Olga is leading a multidisciplinary team responsible for bioanalyticals, supporting Pfizer clinical development portfolio and working with external partners and CROs. The group is responsible for delivering assays and data enabling Pfizer clinical programs. I think there is some, okay. People can see my slide now. The group is responsible for delivering assays.
We can see Olga.
Can you see now?
We need to advance on more.
Yes, go ahead, please.
Okay, is this the current slide?
Next one.
Okay. So her group is responsible for delivering assays and data enabling Pfizer clinical programs and regulatory submissions of biologics, small molecule, and biomarkers across the therapeutic areas. Our second speaker today is Dr. Deanne Jackson-Rudd. Deanne is currently principal scientist at PPDM department at Merck Research Labs. Deanne is currently the scientific lead for the HIV modeling and simulation effort within the quantitative pharmacology and pharmacometrics group. She is also the digital health lead for HIV and schizophrenia projects. Before joining QP2 in 2014, Deanne spent 10 years in Discovery Bioanalytical Group. Without further delay, we begin our presentation. Dr. Olga.
Advance, next slide, please. Okay, so greetings, everyone. Thank you, first of all, thank you for attending this webinar and for your interest to patient-centric sampling and innovation in biospecimen collection. When we think about the innovation and technologies surrounding us every day, it's amazing how much progress have been made over the last few decades. From telephone, photo cameras, paper records, calendars, radio and television, books and libraries, we have moved to a single device, a smartphone, all in one. However, for many years for biospecimen collection, which touches every individual in our day-to-day life, either as a routine blood work or medical diagnostics, sport doping control, and unfortunately, disease monitoring is remaining largely the same. For almost 7 decades, the same venipuncture and containers are typical and the most frequently used technique and device for blood collection. The pharmaceutical industry is not an exception. Participation in clinical trials required blood collection. What is innovation in blood sampling? What are alternatives? What is driving the innovation? Next slide, please.
Okay, I have moved the slide on
Can
my
we get
end.
to the next slide, please?
Yep, I have moved the slides from my end. Apparently, yeah, we're having some issues here.
So we're okay. Thank you. So we are all living in a new reality of COVID-19 pandemic. As we see over the last 3-4 months, This is new reality and necessity of the COVID-19 pandemic is driving the innovation in public health and breakthroughs in ways how the diagnostics tests are conducted and results are delivered back to the doctors and patients. Huge demand and constrained capacity of the health services in this area and need for social distancing and PPEs are driving the implementation of the fundamentally novel approaches in public health. Drive-through testing sites, telemedicine, at-home sample collection, services for ultra-rapid patient sample pickup and delivery to the testing lab and vaccine trials. All these alternatives were implemented extremely rapidly. Next slide, please. Now, the COVID-19 serology testing is coming as very high priority to understand immune response at different phases of the disease and its epidemiology. SARS-CoV-2 antibodies identification within the population is a key step to detect and inform who has been infected by COVID-19 and produce antibodies in response of the infection. Those tests will be able to show how many people have had the disease. Individuals, including individuals experienced very minor common cold-like symptoms or even were asymptomatic. Understanding of the epidemiology and real spread of the virus is critical for managing the pandemic and respond appropriately to the potential new outbreaks. That research would require a large observational sero-survey study in healthy population. How to conduct it in conditions of social distancing and elevated risk for medical personnel. National Institute for Health initiated in an observational study to quantify undetected cases of coronavirus infection. SARS-CoV-2 pandemic sero-survey and blood sampling study is recruiting now the participants. NIH researchers are targeting to enroll about 50,000 volunteers and collect and analyze blood samples using either blood collection at Bethesda NIH campus, primarily for the employers who would like to participate, or mostly at home, using Neoterix meter home blood collection kit for at-home blood sampling and self-collection. Another example of remote sampling approach using different collection device is HEMA6 DB10 for COVID-19 serology testing. Same purpose, different approach for selection of the collection device. This device enables the collection of the control volume of blood from a simple prick at the patient's fingertip using patented microfluid technology sampling. And Joanne will talk more about the device later in the presentation. Next slide, please. So I'll start talking while waiting for the slide changing.
Yeah,
the innovative break. No, it's not yet moving. So
yeah, I think Maha, you have the control. Is there a way you can give me the control? The slides are not moving.
There you go. Thank you. So the innovative breakthroughs driven by the COVID-19 pandemic will tremendously accelerate implementation of patient centricity in pharmaceutical industry. New digital and remote sampling technologies, virtual clinical visits, mobile phlebotomy, etc., will transform clinical trial conduct. Clinical trials will come to patients' home as a part of new normal. Next slide, please. So when we're talking about patient-centric sampling in pharmaceutical industry in 2020, it's important to highlight what we already know and what yet to be learned and understood. We know it can be done, and I will share some good examples of success in various areas. We understand the clear benefits to the patients: less pain, reduced burden of clinical sites or lab visits, more flexibility for ambulatory treated patients, and less stress for the family with elderly and pediatric patients. How does it benefit the organization and pharma? What is the path forward for more rapid implementation? What is on the way? How could we leverage our communities? Many questions to ask. And let's discuss some of the sampling options and examples. Next slide, please. Historical perspective, dry blood spot. I would like to pay tribute to pioneer technology, which is well known these days. Many of you are familiar with DBS and have experience using the relatively old technology in your clinical studies. Pediatric diagnostics is using it for decades. This slide is just a reminder of our starting point and challenges associated with hematocrit and DBS sample quality and consistency. Next slide, please. The next step forward was using dry blood for analysis was volumetrically controlled blood collection, which has which was designed to alleviate a hematocrit challenge. Initially introduced by Phenomenax, the tips absorbing small and fixed blood volume was targeting major improvement in animal pharmacology and toxicology studies, especially in small rodents, to enable serial sampling. Clinical applications and clear demand for an alternative blood collection led to further development and commercializations of microvamps by Neoterics, which allowed to overcome hematocrit factor and simplify the collection process and provide better sample quality. It was a step forward in at-home collection. Next slide, please. How microvamps been used? The very good example of research study conducted by Professor Christoph Stoll and his team at Ghent University. In this research study, phosphatidylethanol, a direct alcohol marker of the alcohol amount consumed in the previous months, was measured in blood of the study participants. Sampling strategy, 3-finger prick and blood absorbed on microvamps tip over 1 month, mailed back to the university lab. Enrollment, very flexible. You don't need to go anywhere. The participants were enrolled by email, telephone. They received trainings over the hard copy leaflets and video. And taking home message, flexible remote enrollment and at-home blood sample collection using microvamps collection tips resulted in a record rapid recruitment of about 800 participants in this academic research study. This is an example how patient-centric approach may transform clinical studies. Next slide, please. Another good example of successful implementation of the blood collection device developed and manufactured by 7Sense Biosystems, touch-activated phlebotomy app for protein biomarker analysis was demonstrated by Novartis Institute for Biomedical Research, the biomarker team. This graph is showing a perfect, I would say, perfect CRP correlations, concentration correlation between tap collected blood and traditional venous blood collection. Slide graciously shared by Dmitry Mikhailov from Novartis. Next slide, please. Another example, TASA on demand is an alternative blood collection device equipped with either removable cartridge loaded with 4 volumetric absorptive microsampling tip for dry blood collection, or alternatively equipped with plasma or serum collection microtube. Implemented in a clinical trial, this technology will reduce patient burden and enable patient participation in clinical trials using at-home collection. And one perfect application for that, if you wish, is the participation of the patients enrolled by patients with muscular dystrophy, which hits young boys at around age 4, worsens quickly, and life expectancy is not that great, so like 20s. This is a rare disease, and the patients do not have cure at this point. It's extremely painful and hard for DMB boys to provide venipuncture or conventional blood samples, and difficult for families and kids to get to the clinical sites. TASA device offers this option and alternative possibility for near-patient sampling combined with painless literally painted sampling. The plan is to conduct phase 1B study in 2020 to compare conventional blood PK samples with TASA samples sampling conducted by medical professionals, and move forward with potential use of the device and later clinical trials in DMD boys. Next slide, please. Another area of the application is with huge growth potential is clinical lab testing, conducted remotely to support physician-prescribed or decentralized clinical trial, clinical laboratory tests, for example, lipid panels, liver function, diabetes diagnostics, etc., many more, would reduce need for clinical sites or local diagnostics labs visits. Laboratory test application using the self-collection device developed by Boston Microfluidics, BMF, was developed by LabCorp and presented by Russ Grant at Land O'Lakes Analytical Conference in 2019. The workflow includes elution extraction from the device and either automated analytic analyzer or LCMS detector could be used for analysis. You can only imagine how much easier would be for severely ill people with limited mobility to do the basic and oftentimes critical laboratory tests using this approach. Next slide, please. Less invasive approaches are not only limited to alternative collection techniques and devices, but also exploring using the different and sometimes non-traditional matrices. It's not all about blood. The study conducted in Nepal by the University of Groningen, Netherlands, Nepal Ministry of Health, and University of Sydney demonstrated that saliva levofloxacin levels correlated with paired venous blood collection in 23 patients enrolled with multi-drug resistance to tuberculosis. Another example of evaluation of the adherence is evaluation of the adherence to antiretroviral drug by HIV infected patients using their hair. Literature review of testing by measuring concentrations of the drug in a level antiretroviral drugs in 31 studies and HIV patients suggested, not perfect correlation, but suggested that hair concentration of the drug is a valid biomarker of antiretroviral medicine adherence. Pharmaceutical industry, diagnostics, medical community, and the most importantly, patients need less invasive, less painful, and absolutely flexible biospecimen collection, which would enable quality data, support medical decisions, and would bring patient benefit to patients. And Diane, take it to the next topics of the presentation.
Thanks so much, Olga. As shown in the examples provided, we believe the benefits to the participant are high. There are also benefits to the organizations who use these devices in their clinical trials. These devices represent the first step in a paradigm shift. Currently, clinical trials are site-centric. This leads to geographical limitations, participant burden, data loss between visits, and limited data. Site centricity also leads to operational inefficiencies like transcription errors, data management that is laborious, and paying for many clinical visits. On the back end, we really don't know how much drug is taken by each participant and when they're taking the drug. These inaccuracies have an impact on dose selection and exposure response calculation. Next slide, please. By shifting to patient-centric sampling, we can move the needle. At-home sampling allows people in more remote locations to participate in trials, lowering the patient burden. It also means you can collect data between visits, which allows a more complete view of the participant in the trial. More PK data allow for a more robust exposure response analysis and can help teams understand trial failures. When clinical trial procedures are linked digitally, there is less burden on data management and our reconciliation team. Also, when linked to other digital health solutions, patient-centric sampling can help us know when and how much drug is taken, which leads to a better understanding of the relationship between drug exposure and efficacy. While the impact that patient-centric sampling coupled with things like smart packaging or electronic adherence monitoring to PK/PD modeling is not fully understood at this time, it is clear that better, more accurate data could be extremely beneficial to our organization. Next slide, please. When can patient-centric sampling specifically help an organization? This is a small molecule example where, due to compound characteristics, there was high red blood cell binding. A liquid whole blood assay was cumbersome, and so the team considered dried blood vein sampling as an alternative approach. In the plot on the left, liquid blood concentration on the y-axis is plotted against veins dried blood concentration on the x-axis. As you can see, the correlation is quite good. The plot on the right shows the concentration time course for plasma in green and blood in red and blue for liquid blood and dried veins blood. The liquid blood and veins blood concentrations overlay. The team was also able to leverage this work and the long half-life of the molecule to enable at-home PK collection. Next slide, please. Patient-centric sampling is a pretty obvious choice for pediatric participants, but there are also organizational benefits. In this example, the Tasso on-demand device was leveraged to get intensive microsamples in juvenile idiopathic arthritis patients to bridge PK to adults. This intensive sampling would not have been possible with traditional sampling due to blood volume restrictions. Next slide, please. In order to move to a more patient-centric approach, we need tools. And Olga has mentioned several of those through the examples, and I'll give a little bit more detail about some of them. There are several ways to take blood samples that allow for patient-centric sampling. Dried blood spots, or DBS, have been used in both a clinical and at-home setting for many years. Dried blood spots are collected from a finger stick, followed by dabbing one's blood onto a paper card. This is shown in the image on the far left of the slide. While DBS works well in certain settings, there are sometimes issues with participants not being able to spot the card reliably. Several companies have developed technologies that improve upon the DBS example, but still require a finger stick to access the participant's blood. Second from the left is the Neoteryx Mitra volumetric absorptive material sample, or VAMES, which was mentioned in the high red blood cell binding example. Also employing a finger stick, the VAMES system absorbs a prescribed amount of blood on a tip. This removes the inaccuracy of spotting blood onto a paper card and ensures that the bioanalytical group will get a precise amount of blood per sample. In the middle of the slide is the HemaPen, which is a microsampling tool that enables the collection of 4 volumetrically fixed, accurate and precise samples from a single source. The Hemaxis is shown top right, is a microblood sampling platform that uses microfluidics to collect and prepare accurate volume-controlled samples of whole blood, plasma, and serum. And finally, shown on the bottom right, the Capitainer QDBS provides convenient and quantitative blood sampling at the fingertip for everyone, everywhere, at any time. This enables easy and accurate sampling by non-healthcare professionals. Next slide, please. There are some participants who are not entirely thrilled about sticking their finger for a blood sample, myself included. For those of us who are a bit squeamish, there are companies who are developing devices that access blood from the upper arm that are touted as pain-free or nearly pain-free. Their aim is to not only make blood collection less painful, but also make it simple enough to do at home. Further developments in these devices are needed to be truly transformative, including collecting things like time and date stamps. Also, collecting data like ambient temperature could be useful for more sensitive samples. The 7th Sense Biosystems TAP, shown on the left-hand side of the slide, and the Tasso on-demand device, shown on the right-hand side of the slide, collect blood, typically from the upper arm, through a virtually painless process. There are similar devices being developed by Loop Medical and Drawbridge Health. Next slide, please. Great. So we have all these options for how to collect blood in a patient-centric way. But an important thing to remember is that without a reliable bioanalytical assay, the concentration data will not be accessible. Our bioanalytical groups work through many aspects to build a reliable assay. Is the mass spec sensitive enough to pick up the compound of interest in such a small sample? Is the compound stable in the dried state? Can the compound be reliably extracted when these samples have been through high heat or high humidity? Is there automation available to help with handling these samples? Are there other things that impact these samples, like lot-to-lot variability of the device? Our bioanalytical groups take all of these things into account when determining if they can provide high-quality data to the project team. This is all so much more complicated than developing an assay for a traditional plasma sample. Due to the complications involved in determining the viability of patient-centric sampling from a BA perspective, it's important to have team discussions early and often as your program develops. Next slide, please. So, assuming your bioanalytical group can work their magic and feel that high-quality data from a patient-centric sampling are feasible, are there other things that might make the road difficult? These techniques are new, so site and patient training on how to use the device are important. Some of these devices may require other technologies as a partner, such as smartphones. This can cause concerns in remote geographies. Some of these companies are new and still learning about how to provide the number of devices we need with the consistency we require. While providing further complexity, especially from a validation and data management perspective, having the device not only collect blood, but also time and date at which the sample was taken would be awesome. It's also recognized that there are some patient populations that may feel that at-home sampling is not appropriate for them. We also, as with so many aspects of a clinical trial, rely on the patients to comply and help us collect reliable samples. Just like with the complexities encountered from the BA perspective, due to these logistical considerations, team discussion is quite important. Next slide, please. New tools are fun and cool, but they do increase the cost of your trial and the complexity of your protocol. They may also impact enrollment. While regulators are excited about the opportunities that these devices can provide, they caution that there are many steps to consider. Have you adequately proven that the concentrations from dried blood are bridged to liquid plasma? Have you considered the path to importing the device into each country running your trial and any local regulatory approvals needed? How do you know if an at-home sample is from the participant enrolled in the trial? We are in the early days of understanding how to implement patient-centric sampling and what the return on investment might be. There's no playbook and there are no definitive data. But despite barriers, we feel that patient-centric sampling can help revolutionize the way we do clinical trials. Next slide, please. Moving forward is going to take patience and cooperation. Sharing our successes and failures in groups such as the IQ Working Group, Transcelerate, and PCSIG will help us all move forward. Next slide, please. We've done all of this. The world is so different than it was just a few years ago. Next slide, please. And again. It's time for clinical trial sample collection to follow suit. Patient-centric sampling is the wave of the future. Next slide, please. With patient-centric sampling, we can access novel endpoints, build more predictive models, and understand better the effect of our potential drugs. We need to work through the burden of implementation and regulatory acceptability. We'll get more and better data and increase our access to participants while decreasing their burden. Working together will lead us down the path of efficient, patient-centric drug development. Thank you all for attending our webinar, and we're happy to take any questions.
Thank you, Deanne. I guess there is a question bar. If people have any questions, please enter them in the questionnaire provided. Also, maybe can we turn on the cameras? I do have some questions to begin. Okay, so I can see you. Yeah. So Thank you for presenting, both Olga and Deanne. So I do have some questions. In your opinion, how COVID-19 has influenced overall patient-centric sampling? Who wants to?
I think that the COVID pandemic has shown us that we need to be bringing these techniques to the forefront. It's become quite obvious that there are diseases out there that are going to impact our population to a great extent. Obviously, we're all very impacted right now by COVID-19. And having the flexibility to take samples at home, not only when it's difficult to get to the clinic, but when it's dangerous to get to the clinic, has brought all of this forward. And I think from our team's perspective, we're hoping that this is sort of the push forward we need to really bring all of this technology forward much more quickly.
I just want to add a couple of words to what Deanne said. I fully agree with every single one. What seems to be happening as a result of the epidemic and the pharma industry and regulatory environment, so that the FDA is getting so much more flexible and so much more open to non-standard and sometimes more risky approaches. So what I would like to see happening, it's not only the technology going forward just because of the necessity, but also open mind from our regulatory colleagues and partners to consider what is really critical, what is less critical, and what we could tolerate in an imperfect yet technology and an imperfect yet digital support for those technologies and clinical operational logistics.
Okay. So you showed that, another question I have is, you showed possible challenges for in-home sampling. In your experience, which was your biggest challenge when you did, did own in-home sampling for your project? Anything you can share? I think anything you can share, things we cannot share, but maybe just give an example which you can share with the group.
In my experience, the participants have been enthusiastic. I think there are considerable bioanalytical challenges. Every molecule is different, and understanding those early in your program is imperative. Working with these small companies that are not used to doing clinical trials is also hard. They're not used to sort of the logistics and the rigor at which clinical trials are run, and understanding how to supply devices that are reliable and don't get in the way of running the trial has been challenging. But every step we learn something, and it's getting better and better.
I would like to add to that. So oftentimes those necessities portfolio driven, right, and project needs. And of course, the oncology, pediatrics are the first demand, rare diseases, the first demand clinical teams who wants that. However, typically it comes in mind when you're very late in the stage of the program, and deep understanding what it takes to jump from on-site traditional blood collection from VIN to at-home collection, peripheral blood using the self-collection device. This is one of the challenges now. So it's education and understanding. And I would love to offer this option to every single team and my company who wants to use it. However, the bridging is not yet there, and it's not always possible because of the analytical obstacles which prevents us from using anything but stabilized plasma or serum. Volume of blood is also critical if we're looking at the biologic implementation. So sampling at home may be limited to the volume. But again, so I don't want to talk about all those obstacles. It's a matter of get breakthrough.
Yeah. Okay. The other question actually came up. There are so many devices. How do you choose the device? And there is also a question on here, and how expensive are these devices?
Devices vary a lot, and the cost of the devices varies a lot as well. In our experience, they haven't been so expensive that they are an overwhelming piece of the overall clinical trial budget. But of course, that depends on the size of your trial, the number of participants, the number of samples, etc. As I mentioned in the webinar, I am a super squeamish person, and I would my personal feeling is that the devices that don't involve a finger stick are really revolutionary, but they're more complicated. And so I think it's really about matching the needs of the program, the needs of the compound, and or what the compound suggests you should use, and the needs of the participants. It depends a lot on disease area and different things, whether people would be all in to do finger stick or not. Olga, did you want to add to that?
I don't have practical knowledge, but I've learned from my partners across the industry. The choice of device, I'm very pragmatic, right? So you may have very cheap choice and not necessarily a complex device, and then like capillary collection in the pediatrics in the hospital setting, and there are projects and programs and drugs like that, right? So you don't use it at home, you use it at the hospital. This is a straightforward path for microsampling instead of more complicated devices. You don't need to dismiss that option. I have my preferences just because I see the slickiness and smartness of devices, and I don't want to talk about specifically of that. But the key to me, what is difficult to get through, and where we focus on, well, first find a partner, find the champions in your company, and find the money, because that costs money. Anything around the implementation of the patient-centric sampling requires funding. And they're not small money, so sometimes you just need to pay the clinical study to bridge. Small clinical study, but yet still clinical study. And cost of logistics as well. I think there will be a strong competition between pharma going after those devices and diagnostics going after those devices too. So we'll see how we can help each other.
Okay. Thank you. So I'm going to read, there are lots of questions coming in now. So I'm going to just read one question which just came in. What is the general perspective of the authorities regarding the acceptance of such technologies? How much efforts have to be made beside the analytical aspect to provide that the right patient has taken the right sample at the right time? Is a digital diary enough, or do we need more sophisticated digital technologies to prove it? Okay.
Great question. I think the regulatory authorities are trying to come along with us in understanding the best uses of the devices. And it's definitely a concern to understand whether the person who's enrolled in the trial is actually the person giving you the sample. And I think there are some interesting ways that you can understand this or combat this. One is you could do a little bit of DNA profiling, maybe spot profiling, to see if there are any weird issues going on. I think one possibility, depending on how big your trial is and how many samples you're getting, is to allow it to be not a requirement of the trial, that participants can opt out if they don't feel comfortable doing it. Because really what you're looking for is people who are excited about being in a trial and complying with all of the aspects of being in the trial. So if for some reason they are not willing to give you an at-home sample when they would be willing to give you an in-clinic sample, I think it's important to understand why that would be. So, you know, we have a long way to go in doing all of these things. I think for some of the device companies, they are thinking about how to couple sampling with some sort of personalized digital stamp so that you have a greater assurance that the trial participant is providing the sample.
Mm-hmm. Yeah, so the digital support for those technologies is crucial as well. So just imagine, once you have collected sample using this device automatically becomes a sample. So which means that you have to apply all the rigor and being able to sustain the chain of custody, including timestamp, right? So including the temperature and humidity control if it's about the dry matrix, dry blood. Also, more sophisticated ways of collecting, well, more sophisticated, including the mini centrifuge at home, so which is actually the prototypes are available, and they're fantastically sleek and nice and light and can be mailed and reused in some way, which can come with a PK kit to the patient's home. So it's more complicated technology. That's right, but it also would allow to collect more traditional matrix plasma or serum, which will be easier to use as a PK sample or lab test samples, because it's already been established early in the trial. It's through the traditional venipuncture. So the bridging is only between the site of collections. You don't need to bridge dry blood with plasma analysis. So it's doable, but it requires extremely much more effort. So I think that the more complex approaches in the technology ways, so it may not be actually collection device. It's already pretty good and performing well. It's a matter of how we can couple it with digital technologies and whatever is available, smartphones for maybe facial recognition, overcoming the patient privacy issues. There are options around, but those options are not yet that mature. So obviously, more complicated technology will not help to facilitate the clinical operations logistics. Getting more complicated, getting more expensive. The clinical trial may turn into more expensive, being more expensive than less.
Yep, and I'm sure that technologies will further develop as we start using these devices more in our trials. So there is another interesting question which came up saying, is there a way to work this novel device manufacturer so there is a collaborative effort to validate routine assays such as the safety labs, common biomarkers, etc., especially when needing to validate values that are outside the normal range, which we typically observe during clinical trials? So what are your thoughts on collaborating with these different manufacturers?
I know that some of the manufacturers are thinking about safety labs. Obviously, that's a huge market for them. And from their perspective, being able to provide their device for standard labs would open up a huge market for them. I don't know that the pharmaceutical industry necessarily needs to collaborate with them on that. I think they're taking that and running with it on their own. Obviously, we can benefit from it. Our group has talked a lot about the fact that being able to take PK samples and biomarker samples and things like that is great at home. But really being able to reduce clinical visits, if you could take safety labs and PK at home, that would be really revolutionary. So it's definitely on several companies' radar, and I think we should keep an eye on it for sure.
Okay.
I agree with that. So, and it's probably a safety labs collection of serum, room temperature storage would be a little less complicated to move forward, except for one thing. So every test will require the individual approval as of now.
All right. So there's another question which came up more towards BioA. Can you comment more on flexibility of these devices or challenges to establish BA method? For example, if we have a compound which requires acidified samples, typically seen in compounds metabolites into glucuronides, what do we do?
I won't get into the specifics, but it's definitely molecule dependent. We have seen compounds that behave very well in plasma with great stability that don't need any special handling instructions that just don't appear to be stable on the absorptive material that we've looked at. So, you know, I think early on in your clinical development, or even maybe before clinical development, if patient-centric sampling is something that the team is interested in, paper exercises can be done to see if the compound is sensitive enough to allow these small samples. And the reality is it's just not going to work for everything. And Olga, do you want to add to that?
Yeah, with all my bioanalytical experience, I just wish we work with the beautiful molecules. Simple, not metabolizing, right? So single peak and a mass spec, and perfect stability, so to handle in room and shape at room temperature. It's not the reality. Right? So we have to look at the what is actually doable from analytical perspectives, because as Diane said, so if there is no quality data or ability to generate quality data based on the sample we have and technologies we have currently, there is no point of investing in this type of complex technology. So we do the best. We draw 5 ml of blood and split them up, and we're adding different additives to different aliquots, so just generate the data. And this is a constraint of the drug development based on just entity we deal with. So what is interesting to look is how much we could use this approach for example, for immunogenicity monitoring. I wish we could step much faster there, but the volume may be constrained unless we collect 2 or 3 devices at a time. That is an area which is very interesting and probably less complicated with respect to the stability or analytics. So that may be a next field to move forward faster. Of course, talking about analytics around that, it's expensive, right? So it's expensive because you have to develop and more complicated assays with much more manual sample processing. Essentially, it takes hands to open up the cartridge and pull the tip out of it. If we just were talking about TARSO, the organization which have the internal bioanalytical groups, bless them, so they can invest into that. So the team like mine, which is working with external CROs, we just need funding. So it's a matter of money and the dedication of the commitment of the organization to and understanding that it does cost more. So it will be investment in the beginning. If we have the durability shown, we can move forward with the program. So those risks, program may not go beyond the POC, but that takes investment in the beginning. For analytics specifically, it's the biggest investment in the beginning of the program. It's analytics.
Okay, so I'll just have one more last question because there are a few more questions, but I think we are running out of time. Last question is more like ClinPharm question. Is it necessary to verify the correlations of analysis using the devices against traditional vein sampling? In other words, is it always necessary to have vein sampling concentration results as the reference?
I think it depends on the device you're looking for or looking at. Certainly, bridging to traditional blood or plasma is important. I think there are some cases where small differences in the site at which the sample was taken are evident, but mostly that seems to be not a huge issue. So I think these are things that groups like the IQ group would like to work through, is exactly what is a good bridge? What does a strong bridge look like? and try to work with the FDA, work with other regulatory agencies to define what that looks like. We know what that looks like for a good plasma sample, what a good BA assay looks like, how we can know we can trust the data. And so working towards understanding that is really important. I think going through the BAMES route is not necessarily necessary, depending on which device you're eventually hoping to use. So yeah, it's sort of it depends on what your end game is, sort of answer.
So I think that option of using more and more the mobile phlebotomy and services at home collection, which the certified nurses will come to collect blood, that could give a perfect opportunity to bridge immediately, because they also can collect at the same time using the peripheral blood collection device. So that's simple and rapid bridging in patient circulation, which has, I think, it has great future. And it's simple.
Thank you both. Maha, do you want to wrap up?